Hello, my dear students. Welcome you all to Sri Jain the Rank Zone classes. I'm your mentor and your friend for the subject biology, and I will be taking the zoology part. Today, we are going to start with MET, that is magnetoencephalography. Encephalography means the study, scanning, or imaging of brain. Okay. So it is a magnetoencephalographic technique which employs squid for picking up weak magnetic waves emanating from brain. So the brain, it emits some weak magnetic waves, and these magnetic waves are captured for production of a digital image by the process that is magnetoencephalography, okay, in which the technique used is superconducting quantum interference device, that is SQUID. Okay, this technique gives information about the health of various parts of brain. So it is totally brain imaging technique. SQUID, superconducting quantum interference device. It is superconducting quantum interference device, example magnetoencephalograph. The device has a superconducting ring where a change which helps him to change the magnetic flux into the voltage in amplified form. So this superconducting quantum interference device, it helps to capture the weak magnetic waves. As the weak magnetic waves, they interfere, they cross each other, they merge with each other. So because of this merging, the amplification of sound waves, amplification of magnetic waves happen. And this superconducting ring, it helps to capture these weak potential waves, and these weak potential waves are changed into a digital image, right? So the weak potential magnetic flux or magnetic waves is amplified to produce pixels or a digital image. Okay, we change the magnetic flux into the voltage in amplified form. Okay, so here basically the magnetic field is, or magnetic energy is converted into the uh, electrical form, or the form of the voltage. The superconducting quantum interference device. Okay, magnetoencephalography. Now we are going to start with biochemical investigation. Biochemical investigation involves the investigation or finding out the abnormality related to the biochemistry, okay, like level of enzymes, level of hormones, and many other factors. So hemoglobin estimation. First, we are going to start with HB estimation, hemoglobin estimation. What is the method of hemoglobin estimation? Hemoglobin, you know, it is a metalloprotein, it is a chromoprotein consisting metalloprotein consisting of the colorless globin molecule. It consists of a colorless globin molecule attached to four red colored heme molecules. So hemoglobin is a chromoprotein, it is a metalloprotein which consists of a colorless globin molecule, which is a protein, and which is attached to the pigment, mean a red colored iron conjugated pigment, that is four red colored heme molecules. The globin molecules consist of two alpha polypeptide chains and two beta polypeptide chains. Okay, I mean, if we talk about the adult hemoglobin, the HbA type, adult hemoglobin. So alpha 2, beta 2. Okay, two alpha polypeptide chains and two beta polypeptide. chains. Heme is a metal complex containing an iron atom in the center of the porphyrin structure. The detailed structure will be given when we will study the circulatory system. Okay, all the tissues I have made you clear. The heme is a metal complex containing an iron atom in the center of the porphyrin structure. Okay, hemoglobin is formed in developing. It is formed in developing erythrocytes, normoblast in the marrow. So hemoglobin is formed in developing the erythrocytes, normoblast in the bone marrow. It is bone marrow. Bone marrow. Okay, bone marrow. So when we are talking about hemopoiesis, process of formation of blood, the hemoglobin is formed in developing erythrocytes, normoblasts in the bone marrow, hemopoiesis. The biosynthesis of hemoglobin involves a triad, mean three things. First is manufacturing of this pigment heme, then manufacturing of globin chains and iron metabolism. So these three things are there. First is manufacturing of heme, manufacturing of globin, and the formation or the iron metabolism. Iron metabolism simply means the iron conjugated proteins, especially the ferritin, because free iron is none of use. I mean, it is not of any use. This free iron, so we have to basically, when we are the iron present in our body is in the form of ferritin, or it's a conjugated iron. It is a complex iron with some ligand. Okay, so methods of estimation of hemoglobin are as follows. Sahil's acid Hamilton method. First is the Sahil's acid Hamilton method. Only I think we are going to discuss only this method. Sahil's acid Hamilton method. Principle is hemoglobin is converted into acid hematin. Hemoglobin is converted, HB is converted into acid hematin. Acid hematin by hydrochloric acid, by treating with hydrochloric acid. The brown color of the compound is matched against the brown glass standard in a comparator. Okay. The compound which is formed, acid hematin, it is having a brown color, and the brown color is matched with the standard glass which is taken in a comparator. So in a comparator, we are going to match the glass which is having brown color with the brown color of the compound, right? This is the heme estimation. This is the hemoglobinometer, which helps to find out the percentage of hemoglobin. The percentage of hemoglobin is calculated in the gram percent. Okay. Normal range of hemoglobin is adult 15.5. Okay, plus minus 2.5 gram per deciliter. Okay, gram percent or gram per deciliter. One of the common method employed to determine the hemoglobin is Saheli method using the hemoglobinometer, brown complex acid hematin method. Some indicators of disorders are if there is low hemoglobin, when for example maybe the condition is anemia or leukemia. If high hemoglobin, mean dehydration or polycythemia or erythrocytosis. Okay, so there can be condition of polycythemia or erythrocytosis or dehydration or hypovolemia. Hypovolemia. Or there can be the condition of anemia or leukemia if there is low HB. So the adult is having 15.5 plus minus 2.5 gram per deciliter. Adult males, they have 13.5, ranging from 13.5 or 13.6 to 16 gram per deciliter. Okay, some book says 14 to 16, 17 gram per deciliter. So there is no perfect range. There is always an average. Average. Females, they have 11 to 13 gram per deciliter, or we can say 13 to 15 gram per deciliter. So in females, the percentage hemoglobin is less in comparison to the males because of the regular menstrual cycle, right? So this is about the hemoglobin estimation. Now coming to next slide. What is ESR? Very important. Erythrocyte sedimentation rate. It is the rate at which the erythrocytes or RBC, red blood corpuscles, sediment. Mean they make a Rolex formation. They get sedimented. When they are allowed to settle down to when they are allowed to settle out from plasma, ESR is measured in terms of millimeter per hour. Okay, that how much Rolex is they are forming. Okay, millimeter per hour. It is useful in the diagnosis of many diseases, though it is not specific for any particular disease. It is useful in the diagnosis of many diseases, though it is not specific for any particular disease. Normal value of erythrocyte sedimentation rate in various methods are as follows. The Winthrop tubes, it says the normal values for men is 0 to 15, okay, and for women is 0 to 20. Okay, it is the settling rate of the RBC millimeter per hour. For example, the RBCs, they are settling down, covering an area in millimeter, and the time taken to settle is per hour. Okay, so this much millimeter of area of RBCs are settling down per hour. Getting me? Okay. So according to Winthrop's tube, it is around 0 to 15 in case of males and women 0 to 20. Western green 0 to 15, women 0 to 20. Okay, so this is a Western green or Westergen method. Okay, or a Winthrop's tube. Methods are almost same. There are some changes. Okay, you don't have to go to the methods. Okay, just remember the name Winthrop's tube. That's important. Okay. So it is the rate at which the erythrocytes are sedimented, RBCs are sedimented to form a Rolex formation when they are allowed to settle out from the plasma. ESR is measured in terms of millimeter per hour. It is useful in the diagnosis of many diseases, though it is not specific for a particular disease. Normal value of ESR is various. Methods are as follows, like Winthrop 0 to 15, 0 to 20, Westergreen 0 to 15, 0 to 20. Okay. Estimation of blood groups. Blood groups detection is termed as a typing of blood, okay, and is generally carried out by the slide technique using anti-ser of A and anti-ser of B. Anti-ser of A means anti-B and anti-A. Antibody against B, antibody against A. Climbing of cells due to serum indicate the blood groups as given below. It is not climbing, it is clumping. It is clumping. Clumping of cells due to serum indicate the blood groups as given below. Okay. So, the blood group O, it is having both the antibodies anti-A and anti-B. Okay. Blood group A is having anti-B but not anti-A. Okay. Blood group B is having anti-B, not anti-A. AB is having both the, means AB is having both the antigens. Both the antigens. B is positive for antigen B. A is having antigen A, and O is not having any antigen. O is having both the antibodies. Both the antibodies. But no antigen. Okay. So anti-ser of A and anti-ser of B is taken to treat the RBC. Okay. When the A blood group is treated with the anti-ser of A, it is, see, this is sera. Sera means we are talking about the antigen. Okay. And if we take anti-sera, then it becomes an antibody, right? So if it is sera antigen, then blood group O is having sera, no. Both the seras are absent, whether it is anti-A, whether it is anti-B. Understood? A is having sera for A, but B is absent. That is why given. B is having sera for B, A is absent. And AB is having sera for both, right? Now comes the lipid profile test or the blood cholesterol test. Okay, to measure the triglyceride levels. Okay, measure the levels of lipids, fatty acids, cholesterol, triglycerides in the blood. Elevated lipid levels indicate the heart diseases or risk of heart disease increases or stroke, overweight or diabetes mellitus. To measure the level of lipids, fatty acids, cholesterol and triglycerides in the blood. Elevated lipid levels indicate heart diseases, stroke, overweight and diabetes mellitus. Okay. Two important cholesterol types are high density lipoproteins, low density lipoproteins. Okay. High density lipoprotein have a protective effect against the atherosclerosis, whereas the high blood level of LDL is a risk factor. Low density lipoproteins, it is considered as a bad cholesterol. And high density lipoproteins, it is considered as good cholesterol. So two important cholesterol types are HDL high density lipoprotein, LDL low density lipoprotein. HDL has a protective effect against atherosclerosis, okay, whereas the high blood level of LDL is a risk factor. Okay, adult 15.5 to 2.5 gram per deciliter, okay, plus minus. Normal cholesterol level in blood is 150 milligram to 250 milligram per deciliter. High risk levels is greater than 240 milligram per deciliter. Okay, normal cholesterol level in blood 150 milligrams to 240 milligrams per deciliter, and high risk levels if it increases more than 240 milligram per deciliter. So a lipid profile test is done to check out different types of lipids. Okay, like LDL, low density, high density, triglycerides, and chylomicrons. Right? This is nothing. I think this is misprinted. Okay, so don't consider it. Normal cholesterol level in blood is 150 milligram to 240 milligram per deciliter, and more than 240 is a risk factor. Okay, it induces or it imposes a potential risk. Right? So this is the lipid profile test. Now coming to next, ELISA test, enzyme linked immunosorbent assay test, or it is a diagnostic kit which helps to determine the complement formation or antigen antibody reaction. It is a technique which can detect and even quantitate extremely small amount of protein. Okay, small amount of protein in the form of enzyme, a toxin, antigenic form, etc. Right? Antibody or antigen with the help of enzyme. The reaction is allowed to proceed in a specialized ELISA plate or tray. Okay. The commonly used enzyme are peroxidases and alkaline phosphatases. Okay. The commonly used enzyme in ELISA are peroxidases and alkaline phosphatases. Okay. ELISA for HIV antibody in serum. ELISA for HIV antibody in serum is used to diagnose or screen the HIV, human immunodeficiency virus. You can see first the test serum plus antibody, they are given. Okay, when the serum which is taken for examination is added with the antibody. So the serum which is taken for examination, when it is added with the antibody, then if the serum means the paratope and epitope, they mix, they form a complement. Okay. So the antigen becomes immobilized. Antigen becomes immobilized. Then anti-human immunoglobulin coupled to enzyme peroxidase. Then anti-human immunoglobulin, okay, whether it is IgG, IgM. So against IgG, IgM, the anti-human immunoglobulin is coupled to enzyme peroxidase. It is coupled to enzyme peroxidase. Okay. Then chromogens are added like OPD, HCl, PN, PP. Mean OPD, HCl, paranitro, phenol, okay, or these are the chromogens. These chromogens, they provide a particular color, and because of the color distinction, it becomes easy to diagnose. It becomes easy to diagnose. Mean to determine whether the particular antigen is matched with a particular antibody. So if a person is suffering from HIV and he or she is carrying the antigen for it or antibody for it, then if we inject the antigen makes a complement with the antibody, right? So ELISA is used for those purposes. A microassay plate which is coated with HIV antigen, it is coated with the HIV antigen. The sample suspected that contain antigen immobilized on the surface of the ELISA plate. Okay, so there is a microassay plate which is having HIV antigen all over it. Then the sample which is suspected to have a immobilized antigen, okay, containing the immobilized antigen on the surface of ELISA plate, means the plate which is having the antigen, it is the antigen is immobilized by giving it the the antibody or the antisera. Okay, so this antisera or antibody makes the antigen immobilized. Okay, the test serum is added and incubated. The test serum is added and incubated. HIV antibody, if present, the serum will attach to the HIV antigen. That's what that means. Both the points are same. Okay. After washing, add goat anti-human immunoglobulin, okay, anti-G or human immunoglobulin. After washing it, adding goat anti-G human immunoglobulin and antibody conjugated with hot serum peroxidase enzyme. Then we complement or we attach the antibody which is conjugated with hot serum peroxidase enzyme. Okay. The conjugate will attach to the HIV antibody, then the conjugate will attach to the HIV antibody in a positive test. If the test is positive, then the conjugate means what? The antibody which is conjugated with peroxidase enzyme. If the antibody after conjugation with peroxidase enzyme, okay, is present, then it is a positive test. So what we have to do, first we have to take a micro assay plate coated with HIV antigen. Then this HIV antigen is bring closer to the antisera. If the antisera makes a bond, paratope, epitope, a lock and key with the required antigen, HIV antigen, and after washing it, we have to check whether the antigen is mobile or it is temporarily immobile. Okay, so after washing we will add the anti-G human immunoglobulin, okay, antibody which is conjugated with the hot serum peroxidase enzyme. If the conjugation happens, then the test is positive. After washing, add substrate OPD, orthophenyl diamine, HCl, hydrochloric acid, and paranitrophenol phosphate. So after washing it, we have to add a substrate like OPD, that is orthophenyl diamine, HCl, hydrochloric acid, and paranitrophenol phosphate. Okay, or hydrogen peroxide along with 4-chloronaphthol. Okay, so these are chromogens basically. These are chromogens. These substrates are chromogens. Okay, chromogens, whether it is OPD, orthophenyl diamine, or paranitrophenol phosphate, okay, or hydrogen peroxide with 4-chloronaphthol. A color develops in the positive test while there will be color in the negative test. Okay, a color develops in the positive test while there will be color in negative test. ELISA is a very quick diagnostic technique for the detection of wide variety of pathogens, disorders, allergens, and hormones like. Mean a positive test develops a color but there is no color. There will be no color in the negative test. Okay, so please rectify it. There is no color in the negative test. ELISA is very quick diagnostic technique for the detection of wide variety of pathogens, disorders, allergens, and hormones. But the fact is, a color can develop to the similar antigenic responses as well. That is why ELISA, it is good for screening. It is good for screening, mean screening. Okay, but for that, but for confirmation we use, but for confirmation we use blotting techniques like southern blotting, western blotting. Okay, because if the same antigenic response is shown by some other human T lymphocyte virus, okay, then it can also happen. So pregnancy test through presence of human chorionic gonadotropin in urine or blood. ELISA is used nowadays. AIDS or HIV infection, hepatitis, STD, sexual transmitted diseases, rubella virus, thyroid disorders. So all they are diagnosed, confirmed, okay, screened as well as confirmed, okay, by this test, okay, ELISA. Auto-analyzer. Auto-analyzer is an automatic computerized instrument which can find qualitatively as well as quantitatively various biochemicals present in the body fluids like urea, uric acid, ketones, cholesterol, glucose, proteins, and enzymes. So it is an automatic computerized instrument which can analyze qualitative as well as quantitative status of the biochemicals which are present in the body fluids like urea, ketones, uric acid, glucose, proteins, and enzymes. In a batch type autoanalyzer, which is the more common type of autoanalyzer, batch type, only one type of analysis is carried out at a time, while in a multi-channel autoanalyzer, a number of biochemicals are tested from the same sample. So a batch type autoanalyzer, which helps to which helps to quantitatively estimate or qualitatively estimate about one biochemical, while the multi-channel autoanalyzer, it can diagnose or it can find out the multiple biochemical status of multiple biochemicals. Polygraphy. It is popularly but quite incorrectly called the lie detector. Okay. Polygraph is a simple, compact, and portable machine that records qualitative changes in physiological parameters rather than measuring them accurately. Okay. So basically, polygraph is a simple, compact, portable machine that records qualitative changes. What are the changes in the quality of biochemicals? Okay, during the physiological reactions, physiological parameters, rather than measuring them accurately. These parameters include vital traces such as cardiac variables, heart pulse rate, relative blood pressure, the rate and depth of breathing, and the resistance of skin to the conduction of electricity. A modern computerized version of this multi-channel device is used to record neurophysiological parameters such as EEG, EMG electromyograph, EOG electrooculograph for eyes, use of polygraphy, lie detecting, monitoring the stages of sleep, okay, find out alpha, beta, theta, delta, all the waves, REM sleep, okay. Then electrophysiological behavior of brain and its functions. Okay, so polygraphy is a machine which helps to diagnose, which helps to create the electrical stimuli, and these electrical stimuli are basically favored by the uh, processes in our body. The physiological processes of the body and their rate of happening basically tells us, or basically digitalized, it gets digitalized, okay, in the form of a graph or anything else which help us to diagnose, okay, the metabolic functions are going well in a condition. Okay, so this is what polygraphy. Laser. Laser is light amplification by stimulated emission radiation, which is a beam of very high energetic particles. Okay, depending upon the source of generation, it is called argon laser, carbon disulfide laser, carbon dioxide laser, sorry, carbon dioxide laser, or neon laser. Neon laser. Medical lasers are used for therapeutic purposes. They can be targeted or any tissue or organ inside the body. So laser is light amplified or light amplification by stimulated emission radiation, which is the beam of very highly energetic particles. These energetic particles, when they strike through the body, so they gets penetrated through the body. Okay, so it is called, I mean, it is of various types: argon laser, carbon dioxide laser, neon laser. Okay, this can be targeted on any tissue or organ inside the body, breaking of the kidney stones, gallbladder stones into fine powder for removal through natural excretory processes instead of surgery. Treatment of cancerous and other tumors by selecting burning of cells. Treatment of opacity of eyelids. Okay, so laser is used to various in various medical fields nowadays, like breaking of kidney stones or gallbladder without any surgery, treatment of cancerous cells without invasive surgical instrumentations, and treatment of opacity of eyelids. Okay, laser microsurgery. In microsurgery, type, use, and effect of laser on the tissue depends upon its wavelength. The type of laser used in microsurgery: carbon dioxide laser. This laser has a short wavelength of 1.6 meter. It is absorbed by water in target cells, and its energy is quickly converted into heat. As a result, the effect of laser is highly localized, used in neurosurgery to remove the inoperable brain tumors. So carbon dioxide laser, it produces a wavelength of around 1.6 meter of wavelength. Okay, wave of wavelength having 1.6 meter. It is absorbed by water in the target cells, and its energy is quickly converted into heat. Okay, as a result, the effect of laser is highly localized, used in neurosurgery to remove inoperable tumors. Visible argon ion laser, it is used in eye surgery, has an intermediate effect on tissue. Okay, visible argon ion laser. Okay, then Lasix laser, it is used to break the kidney stones. Okay, then cryosurgery. Cryosurgery is the cold treatment, as the name implies, using freezing temperature to destroy. Okay, liquid nitrogen, using temperature to destroy what? Different types of infection causing agents or the tumors or the abnormal fat cells. Liquid nitrogen, which has a temperature of minus 196 degrees Celsius, is sprayed onto the tissue either directly, as in the treatment of warts, via hollow probe that is inserted into the tissue. Cancerous tumors can also be destroyed in this way. Okay, so cryosurgery involves the freezing, extreme freezing of liquid nitrogen, freezing temperature of liquid nitrogen to stop the blood flow. Okay, it will stop the blood flow, and those anomalies, overgrowth, warts, okay, or cancer or the other tumors, they can be deployed off. Okay, understood? So this is all about the biomedical, techniques we have discussed. Okay, just go through these techniques. Okay, not all of the techniques are very important, but ELISA is quite very important. The hemoglobin level, the ESR. Okay, so one question can be, it can be easily taken out from these topics. Okay, we will meet in the next lecture. Please revise to these topics because these are the miscellaneous topics, and you doesn't I mean you have to prepare these topics very separately, right? So we'll meet in the next turn and we'll start the new topics again. Till then, bye bye. Take care. Stay safe.